Thank you very much. So we have slides, I think. Yeah, thank you. My name is Lucien Benheim. I'm from Laser Analytica and the London School of Hygiene and Tropical Medicine. We have been working with a large group of scientists and participants from the pharmaceutical industry to try and tackle this question of identifying gaps and signposting solutions. I like signposting. I think that comes from you. For efficacy and effectiveness. So the main goal of this talk would be to give you an overview of the work package 2 work that we've been doing, number one, to describe the decision-making approach that the work package 2 has developed and for research and development. And when developing a new compound, it's very important to say that we've been really trying to focus on developments of new compounds and receive feedback from you, obviously. So should the audience, I'm sorry that I have to look here, but do after seeing the presentation, so we'd like to understand the concepts of the drivers of effectiveness. We've worked on these concepts because it's very important to understand the concepts. If you want to design study, you have to really understand what you mean by effectiveness from an operational viewpoint, and the methods to identify the drivers of effectiveness. So during the development, this term, drivers of effectiveness, was a term that we developed at Laser Analytica before GetReal, and has proven to be very effective when you want to develop and design studies and finally develop your program and for your drug. Be aware of tools and analytical techniques available to support the study designs aiming to assess effectiveness. So I will present to you a number of solutions that we have developed during Work Package 2 and identify the opportunities to apply concepts within their organizations. I don't know how it works. So the main objective of this work package was really to better understand the gap between efficacy and effectiveness. A lot of people talk about the gap between efficacy and effectiveness, and there is this general idea that effectiveness will be lower than efficacy, and it's not true. Actually, when you look at most of the work that has been published, it's rather the contrary. Very often drugs do better in real life than they do in trials because they are targeted to the best population in the practice, whereas the trials are very often using a small sample of the population. The reverse can be true too. You can have situations where effectiveness doesn't come up as good in the real life as efficacy is. But if you want to be able to study that, not only conceptually, but really to do the work and gather the data and put it together to be able to not only measure the effectiveness in the real life, but to predict the effectiveness at the stage of development, and moreover, to be able to implement within your trials at the very early stage the kind of collection of information that you will be needing to have the information to predict the effectiveness after the trials. So a lot of work has to be done. And this is what we have been doing during this work package. We've been trying to work on methods to identify as early as possible and test as early as possible the drivers of effectiveness. And we have come up with a number of toolboxes for this that I will be presenting to you, that Chris and I will be presenting to you very quickly. And we also wanted to focus a little bit on the analytical techniques and methods that could be used in order to actually take the right decisions all along the development of the drugs. So first, what is effectiveness? It is not, as some people think very often, a degraded form of efficacy. Effectiveness may be defined as the impact of drug efficacy when all interactions are at stakes, so are at play. So if you allow all the interactions to actually that may actually have a role or modify the effect of your drug, And if you make it work, then you will see what the real effectiveness of the drug is in the real life. In a sense, you could say that efficacy is effectiveness in a smaller sample of the population, which is the population that has been subtracted from the real world to study a drug. It's just, actually, we would like to reverse the thinking about effectiveness. We have developed in a model that we published before GetReal the idea that there are 3 levels of interactions that are considered, and we have during GetReal tried to really study these levels of interactions. First, the real and the actual effect of the drug itself. Obviously, it has a biological effect, it has a pharmacological effect, so that's one factor that works towards effectiveness. Second, the patients and the disease-related characteristics. And third, the healthcare system itself. And the framework works like this. Oops, sorry. Can I come back? Yeah. So, I don't know if it works. Doesn't, okay. If you can see the red thing. So, very often people concentrate on the health system, and they say that the effectiveness is the drug isn't working or is working differently in the real life because the drug goes out there in the health system. But I think what we have to first concentrate on is what are the factors that modify the effect of the drug on the real life, that the efficacy of the drug on the real life, and we must be able to measure them. And the first type of factors, obviously, will be how the drug is used. What is the actual use of the drug? What doses, what posology, what duration of use, switches, discontinuations, adherence, which is often cited as an important factor, but is not always the only factor and very rarely the most important one. And the co-prescriptions, the past exposure, all these factors of patterns of use will interact with the efficacy of the drug. And it's not to come to effectiveness. And it's very well known. Everybody around this table knows that we do trials with certain doses of drugs, and they are not used at that dose as in the real life. You have to adapt the dose to the patient. This is a very simple interaction, if you wish, for this model. There are many others, not so many, but there are some others, like the switching, the discontinuation patterns, and so on. We will certainly have an effect on the efficacy of the drug towards effectiveness. And you have to measure that, number one. Number two is what are the populations we will receive the drug. There are some disease characteristics, obviously, that will modify the effect of the drug, and some comorbidities. So there are disease-related factors, and there are a number of population factors. I'm an epidemiologist, so I could spend more time, if you wish, on the population factors, such as age, gender, behavioral factors, baseline risk, genetics, that will play a role. All of these factors will play a role in the efficacy of the drug towards its effectiveness. Now, these factors are influenced by the health system, and it is through these factors that the health system that their system will actually interact with the efficacy and produce some effectiveness. The way a drug is covered, if it's covered or not, will obviously change whether a drug is approved. The way the practices of the doctors will be aligned will change the way the drug will be used. And it's through through actual use and through population factors, the distribution of the different factors in the population that at stake, that the drug will actually change the effectiveness would be different from efficacy if it has to be different. So the good news is that there are not so many drivers of effectiveness. Contrary to what people believe or say, if you look at this as this number of factors here, they are not so numerous that we cannot say that we will be tackling all of them for every drug in the future. We have been able to tackle the quality of life of very small populations of patients to be able to calculate the incremental, the ICER, with the right quality of life measurement. And I remember 25 years ago, it was thought as an impossible task. Now we have thousands and thousands of quality of life measurement. We can measure for all drugs. How did these different factors affect the efficacy and towards effectiveness? It's possible to do it, and it's not an impossible task. The second good news, which is very important to understand, very important, is that the interacting factor itself is mostly universal. They will be the same in Africa, in France, in the UK, in Germany, in the sense that age, the way age changes the effect of your drug is going to be the same in most countries. What will change between one country to another is the distribution of the population that will receive your drug. And by changing these distributions, you will, and it's the study of the distribution of the use of the drug that will actually lead you to the measure of effectiveness. During our work, I don't know if it works. So, and the third thing, which is very important to mention, is that you shouldn't confound, and we worked a lot during this work package, interactions and confounding. Very often people will say, oh, when the drugs come to the market, there is channeling bias, it's used different by, this is confounding, which is an issue of importance if you want to measure, remeasure the effect of the drug. Now, when a drug comes on the market, we know it works. We don't have to prove that it works. So we don't have to worry about confounding. We have to worry about the interactions, how the factors change the effect of the drug in the real life, which is not a confounder, which is actually an interaction and is a factor of effectiveness. So we worked a lot during the work package and we have a number of tools to propose to on how to take this into account. I don't know if I'm supposed to stop talking, so no. So one must know what factors are likely, when you launch your drug, one must know what factors are likely to influence the efficacy of the drug, and build your theoretical model about that, obviously. And you have to know the distribution of the factors and how they are likely to affect. Now, the question is how to get an information on this, the effect of age, behavioral factors, disease stage, dose, and so on, before you launch your drug. And, well, there are several ways. I don't know if it's in the next slide. So the purpose of the effectiveness research is actually to assess these interactions. It is to decide which one are universal and which one are really apply only to your drug. And I can tell you in 35 years of experience, I don't believe there are many. How do they distribute locally? And what is the magnitude of their impact? And what is the mechanism of actions by which they may change the efficacy of your drug? And actually see that from a broad perspective. Now, we have proposed during this work package a number of methods for each of that. I will not get into the detail here, and I'm sure that you will come to us. And I think Clémentine is in the room. Clémentine, you're here. So if you have questions, she was the real, actually, person who really made it work with Chris in this work package. So we have developed a number of methods on how to, Clémentine Nordon, so how to identify drivers of effectiveness, how can we address drivers of effectiveness in clinical research. We have developed methods for the use of systematic reviews to identify drivers of effectiveness that may apply to your drug during development. We have developed methods to enrich clinical trials to make them actually more efficient and help you identify some drivers of effectiveness at the same cost and with the same sample size as a regular pivotal study, so without increasing the burden of the pivotal studies. And we have also worked in the work package on how to actually measure these drivers of effectiveness through pragmatic trials. And this is Stuart Van Ster, I think he's here in the room, and he will be answering that questions, and how to use the existing information in databases and also in epidemiological observational studies to identify these drivers of effectiveness. So if you have any questions, you can address and see all these people after the talk. Thank you very much.
Thank you, Lucien. And what I want to take you through is some more of the specifics. So having sort of set out a sort of a framework for effectiveness, how have we tested to try and sort of see how can we systematically assess effectiveness in different disease areas, and how can we turn this into something that's useful for the stakeholders of GetReal. So, is it forwards? Okay, that's not good, is it?
It's even worse for you.
Okay, so is it back, left and right? Okay, so what we've tried to do in terms of putting the output of GetReal into a format that can be useful is to try and provide tools that can help the stakeholders work through what is help to analyze the gap between efficacy and effectiveness. We haven't got the answers to every, how this plays out in every disease area of interest, but what we're trying to do is to show that there's a way of working through this problem that should be adopted. It should be adopted by industry in trying to plan its development program for a new drug, and it should be it should equally be used by the HTA and regulatory authorities when planning for those scientific advice interactions. Because what we'd like to do is to sort of make sure that the conversation about scientific advice is thinking about the effectiveness problem in advance and helping industry understand what its options are and what the acceptability of options are for effectiveness research, not just thinking about how to most quickly get through the registration studies. So the idea is to think about all of these, systematically think about the factors that might cause uncertainty around effectiveness, have a systematic way of understanding those, and then to look at what are the, how that builds us onto possible solutions. So is it possible to identify drivers of effectiveness before launch? And the response to that is yes, it is, and it can be done quite systematically. The methodologies that have been tested in GetReal and will be reported out on the websites over the rest of the year have used systematic or focused literature reviews, expert interviews, have been able to look at an analysis of previous data or to try and reveal differences between efficacy and effectiveness when you have observational data to compare back to trial data. And that can be done by looking at papers, and it can be done by, as I said, looking at individual patient level data. And we've looked at all of those in particular the reports that we'll be putting out and publishing around schizophrenia and Hodgkin's lymphoma, and through this, certain factors, certain factors come out and sort of then are then asking for a solution. In schizophrenia, it was about the duration of disease prior to entry into the trial. So the duration of disease does affect response to drug, and the distribution of that in the trial would be different to the distribution in the real world. So it's one of those factors that you have to understand, and you have to think about how are you going to discuss that interaction when you come to the assessment of the value of a medicine. And as I said, the approaches may be about the trial design, where you're thinking about the population that's represented in the trial, and it could be about the analysis, trying to model and predict the impact of a new drug on that population after launch. I am pushing the button. Okay, so there are a lot of silent builds for some reason. Okay, so the way that we're trying to get through this first question, then how to understand the gap, is to build into our deliverables for widespread use a systematic question-based approach, a framework for working through this discussion. So wherever you are in the phase of development, whether you're thinking about this before your phase 3 design, whether you're thinking about this to work in parallel with phase 3, or whether you're just before launch and thinking, what else can I do to try and address an emerging issue? The first question is, why should I generate real-world evidence? As I come back to this, what is the case to invest in evidence on this drug, on this in our portfolio for this question? So is there a risk of an efficacy-effectiveness gap that would lead to uncertainty in decision-making? And is there a compelling need to generate evidence of effectiveness? And they are slightly different questions. So the first question is about we can help understand the issue systematically, and we're going to put a framework up that sort of helps you work through the identifying the drivers of effectiveness through these different methodologies, the literature review, expert interviews, and data analysis. The use of network meta-analysis incorporating observational data as well as trial data can also help you to reveal how effectiveness and efficacy is played out in the currently available treatments. And qualitative research and patient insight will also give you an insight into things that matter in terms of how behaviors and preferences might affect treatment pathways and outcomes. And the second element of this is to work through this, and very systematically we're going to adopt the PICO approach. So are there questions of efficacy and effectiveness that are related to the patient? Is it the intervention, the way that healthcare is delivered, the healthcare system factors, the availability of resources, the implementation of care pathways, and so on? Are there effectiveness issues around the comparator where the comparator of interest to a decision maker is what's used locally, might not necessarily be licensed, might not necessarily have been tested, might not be in your own trial program. And then the outcome measure is the primary outcome measure for registration. Does it correlate perfectly with the outcome measure of interest to other decision makers and stakeholders? Either does it translate to does your primary endpoint in your trial stand up over time with long-term outcomes? Does it correlate with the clinical endpoint, correlate with economic endpoints? Does it correlate with quality of life and preference endpoints? So having systematically understood that there are ways to look at the question and there are ways of carving up the question, and we will put that framework available to all. And it's a framework that we want. We don't see any reason why that can't be systematically worked through by each company. It can't be systematically adopted by regulatory HTA bodies when they're thinking about how to give scientific advice early on. It gives a structure for all stakeholders. Now, the second question is slightly different. It's one that's a little bit out of scope of IMI, but how can I understand if there's a compelling need to try and address this gap? How do I convince the portfolio review board of my company to invest a few more $1 million in this program rather than a competing program in an equally deserving disease area? And that is where we've been going back to look at previous histories of HTA decisions. Is there a body of case history of HTA decisions? We did this with our COPD workshop. It was a nice piece of work that will be available, I think it's being presented at ISPOR this year, about what have HTA and regulatory bodies had to say about the evidence of efficacy and effectiveness that was presented to them during the assessments of drugs for COPD over, I think it was, Alex, it was at the last 10 years, I think, wasn't it? We had a window of time. And that gives you insights into where effectiveness this evidence of efficacy, effectiveness gap might change and influence the decision. So it's really important factor of understanding the stakeholder's perspective and then putting that back into your R&D decision making. The scientific advice process is going to be absolutely critical going forwards for saying what does that process, how does that process discuss effectiveness, and how does it steer companies towards different options? So a strong scientific advice to do a certain type of program will obviously give you more of a compelling reason to invest in that program. And finally, there's the commercial forecast, which is completely out of scope of IMI. But knowing the impact of the uncertainty around effectiveness, knowing how that will play through HTA recommendations, pricing negotiations, coverage decisions, local access and formulary decisions. Knowing how that will play out will enable a company to work out its commercial hit, as it were, of not addressing the efficacy effectiveness gap. And that does need to be taken into account in R&D decision making, but it is clearly pricing and commercial forecasting are out of scope for IMI, which is focusing on the scientific and stakeholder issues. But but the framework that we're going to be presenting, the tools that we will be making available to people, will steer people through this entire decision process. We need the scientific structure, we need the options, and we need the stakeholder views, and we need to be able to help any team in R&D work through that process. And so when we talk about a navigator, or when we talk about frameworks, this is the structure that we are laying out in a very accessible way for people. So having decided that, yes, there is a compelling need to do something, we need to move on to what are the options and how do we work through what the right options are for us. And again, we can't say now what the right decisions will be for any new drug that's in development. But what we can do is to try and say there are certain types of options which should be considered and which might be suitable for any particular drug development team. So one in particular that's come through over and over again is patient characteristics. There's a lot of work through our case studies has revealed that the populations in clinical trials can be radically different to the population that is then treated immediately after launch. Again, going back to some of the presentations we saw on COPD, it could be as little as 2% of the treated population in COPD are eligible for clinical trials. And the drivers of that are the very strict criteria around trying to get pure COPD and not mixed up COPD asthma patients. So you know that the patient population outside of the trial is going to be quite different in many cases to the clean population that's been tested within the trial. And so one solution of this is to try and include patients you expect to have the drug, include more of them in the clinical trial program for registration. And this is where Lucien and Laser have been working on trying to simulate what would happen to your performance, as it were, the performance of your RCT, if you were to sort of seed it with increasing numbers of patients who represented these atypical trial patients. And I think there's some interesting work that we presented and published this year about you can enrich your trial population. It is possible, rather than just having a black and white exclusion, it is possible to try and bring some of those patients back into trial designs. You won't get fully powered strata, but you will get more information. You'll get a little bit of a toehold from your RCT data that will help you in your predictive modeling. And this is what it's all about. It's not about getting black and white answers here. It's about getting additional information to help with the modeling exercise that is then more acceptable in an HTA process. So we can get more information by seeding and enriching RCTs. Another option that we talked about, the Salford lung study. The other option is to do quite pragmatic designs. And I think the key to this will be confidence about the safety of the drug, confidence to do larger trials with more relaxed criteria. But it is theoretically possible to do very pragmatic designs and start those designs before launch, and have them reporting out in that kind of time window when other stakeholders are making their decisions about coverage and clinical guidelines and what appropriate uptake is. So we're also looking at how methodology of how to perform pragmatic designs. Now, the next talk will be focusing on how we can do that and how feasible and how do we understand how to do pragmatic clinical trials before launch. But some of the methodologies that we've been looking at in our work package have been around the statistical issues of trying to run pragmatical trials in different ways. And one of those is for example, we call TWICs, so trials within cohort studies. So one way of doing a very pragmatic trial would be to try and source patients from a high quality kind of registry or real world cohort to do the randomization and recruitment within that cohort and to do all the data collection through the normal data mechanisms, not to have additional CRFs, additional data collection visits, additional tests. That's a very pragmatic design. It needs the registry infrastructure to be there, which is why the registry initiatives are so important to us. But it has issues of its own about the sampling and the representativeness of different populations if you're trying to recruit in a different way. So, does the likelihood of a patient to consent and continue in a study introduce a new form of bias compared to a pool of patients who were never asked to join the study, or a pool of patients who were asked to join the study but declined to? So, what are the statistical issues around that? And we've gone from the very general into some very specific issues that might help us sort of one by one pick off barriers to doing such research. So the question of, if yes, there's a burning need, what should I do? Our framework on our deliverable will work us then through what are your methodological options for an integrated evidence generation plan? How do you work through what the design features are? And again, depending on whether you're thinking about adjusting your phase 3A, adding with a phase 3B, or thinking about how best to negotiate post-launch commitments, we hope that the work that we're presenting from GetReal will give a kind of a more rigorous structure to what a good study would look like, and what a good study and how a study could answer effectiveness. And I think what we'd like to see is more targeted, more smart designs addressing these questions, and less blanket registry. Please do a registry. We'd like to see much more targeted. Please answer this question, and we'll work together on what the appropriate design is. And again, as I said, the framework will work, depending on where you are in drug development, the framework will steer you towards the work that's been done on pragmatic trials, or the work that's been done on enriched RCTs, the work that's been done on equally importantly, the work that you could do on analysis, network meta-analysis, including observational data and modeling techniques. So we hope to be able to steer in a very structured way a team to work through to what its options are, whether they are studied options or analytical options. And the other work streams in GetReal have either been looking at those trial options, or been looking in a very exciting way, I think, at some of the analytical options, and we'll hear more about that today. So all of the deliverables that we will then be putting out on our website are kind of designed to plug into this. And I've touched on many of them, such as the enriched RCT design, imbalance and selection bias in Twix PCTs. The final point I haven't mentioned on what we've been doing is to look at channeling bias post-launch. And it seems strange at first, because everything we've been doing so far has been, what can we do pre-launch? What can we do before marketing authorization to improve our information? But it's really important to be looking at, well, when we're looking at effectiveness in a real-world population, we've got to be able to try and understand, what will that population look like? How can we predict effectiveness in a population when we don't know what the population will be? We think it will be different to the trial, but we're not, how do we know what it will be? So one of the factors that we have to do is to try and understand from all these different sources, what will the effectiveness population of interest look like, and how does it differ to the trial population? But the other thing is to saying, will that population in the first year be different to the population that's then treated in the second year and the third year? Because we know that there will be the physicians and the agencies that advise the physicians will be steering, will naturally be trying new drugs on different sorts of patients first, and then moving in some way, perhaps, to different populations of drugs, of patients over time. And so we've also been looking at how to understand channeling bias, how to understand the decision-making about who gets what drug after launch, and how that changes over the early years. Because if we can understand that, we can be more, I think, more certain when we're talking about effectiveness, that we're talking about an effectiveness population, that we understand the dynamics of that. So that's how we've tried to pull, and each of these sort of case studies, and more, whether it's the broad framework or the more detailed research will be put onto the GetReal so websites and deliverables and publications and feed its way into the educational materials. 3, 4, 5, 6. I'm half expecting to turn out all the lights at some stage of this. So we hope that after, that what we've given you is food for thought of, understanding, yes, drivers of effectiveness analysis can be done systematically. It is worth doing. It can be incorporated into our early drug development. I think there is a I hope I hope you realise that there is a framework which is equally valuable to the different stakeholders. And if we use a common framework, it's going to help scientific advice conversations. I hope that what we lead to is further conversations around how we can define best practice in doing this. But I think there's more to be done to sort of reach a consensus about on this. And there's some interesting, other groups researching this area. There was an interesting publication from OHE, for example, recently, also on framework of effectiveness, which was very aligned to this. And I'd like to see further research on the drivers of effectiveness, the way that it plays into trial design, the way that it is treated in scientific advice. And I'd like you all to be able to think, how can I take this kind of framework, how can I make it work in my own organization? And I hope that through those of you in the room and those of you and the industry partners within GetReal can all sort of take these sort of learnings back try and internalize them into each organization. So that's the end of our slides. I'm very happy to answer any questions or to field any questions to someone else. Thank you.